The overall goal of this procedure is to demonstrate how adrenal activity can be assessed non-invasively in equines through the extraction and analysis of fecal glucocorticoid metabolites.
This method can help us answer key questions in the area of welfare assessment by looking at the physiological impacts of husbandry protocols on domestic horses, all the way through to wild populations of African zebras.
The main advantage of this technique is it offers a non-invasive option for measuring the long-term adrenal activity in both domestic and free-ranging horses.
On the day of the experiment, after thawing at room temperature, manually homogenize the fecal samples by mixing within each individual sample bag. Then, for each sample, measure out the appropriate amount of feces on a microbalance in a clean weigh boat and transfer the samples into individual 8 ml glass vials. Next, mix 5 ml of 90% methanol in water with each sample and shake the samples overnight on an orbital shaker at room temperature. The following morning, vortex the samples and spin them down. At the end of the centrifugation, decant the methanol fractions into individual 16 by 125 mm glass tubes. Then briefly place the tubes in a 37°C water bath, followed by evaporation of the methanol under air in a fume cupboard. When all of the methanol has dispersed, resuspend the fecal extracts in 1 ml of 100% methanol. Then transfer the extracts into individual 12 by 75 mm polypropylene tubes, cap the tubes,